I'm delighted now to introduce Jamie Lloyd-Smith, who is at UCLA and is going to give us the next talk. So Jamie, do you want to take it away? You're muted. Oh, yes. You're not muted.
Yeah, thank you very much. Let me get myself shared. Great. Okay, well, thank you to the organizers for the chance to be speaking with you. And thank you, Chris, for a very insightful and elegant talk. I think we're going to be odd bedfellows in this session because what I'm going to be talking about is considerably more granular, but does come at some of the same big questions of how to assess the risk of what might be coming and what might be done about it. So I'll be speaking today about transmissibility of emerging pathogens and what we might do to estimate this, to assess risks, and how that can help us control these pathogens. So given that new pathogens largely come to us in the animal world, we think of them in the framework of zoonotic emergence. So these pathogens all begin circulating in animal reservoirs, and they spill over into the human population, at which point they may not transmit at all, or they may exhibit subcritical transmission with a reproductive number less than 1, so chains always die out, or they may have epidemic or pandemic spread with R greater than 1, and we're well familiar with that. For the question of what drives epidemic or pandemic emergence, the crucial thing is a new pathogen comes into the human population and establishes sustained transmission, so supercritical with the reproductive number greater than 1. And it'll be no surprise to anybody attending this talk or listening that human-to-human transmissibility is the very heart of pandemic risk. And this pertains both to its efficiency, so the reproductive number, and particularly whether you find yourself in the sub or supercritical realm. But also COVID has really shown us the routes of transmission need a lot of attention because they immediately mediate possible responses to contain an emerging pandemic. So we want to think about that, and there are two major reasons. So characterizing and quantifying transmission is essential both for risk assessment of possible pandemic threats that have not yet emerged, and this can be all the way to viral prospecting and finding many new possible agents out in the forest, wondering which of these might pose a risk, all the way through to prevention and control once a sort of risky pandemic agent has been identified, or even once a pandemic has begun. And the relative importance of risk assessment versus prevention and control depends where you find yourself across the phases of emergence. So risk assessment is clearly most important for agents that are still out in the reservoir. We're not sure whether they may pose a serious risk among humans yet. As you progress to the right in this diagram to the point where you've already got epidemic spread going on, at that stage you know you have a problem. You don't need to assess whether there's a problem. It's time to pivot to control and mitigation of the pandemic. But what I'll argue, I've been thinking and talking about the risk assessment side of this for some time. COVID, having happened this year, has really revealed that many of the same questions and same approaches that we've been applying to risk assessment have an immediate application once a pandemic is underway, because it is equally important to characterize how transmission works as quickly as possible so that you can target your prevention measures accordingly. So as we think about transmission, it's important to recognize that the effective transmissibility on the ground is really made up of two major building blocks. So there's the inherent human-to-human transmissibility of an agent, just going right down to the biology of how viruses bind receptors and so on. But there's also the susceptibility of the novel host population, or the human population in this case. And there has been an assumption over the years that when a truly novel pathogen emerges in the human population, it will find that population immunologically naive. But increasingly, we're recognizing that this is not always, or maybe even typically, the case. And so really, as we think about the human-to-human transmissibility of a pathogen, we need to bear in mind the two halves of this equation, both the fundamental or sort of R naught transmissibility and the susceptibility of the population. So today I'm going to range over a number of topics and over the years of thinking about these problems. I'll begin with some quick comments on characterizing transmissibility in the field, including thoughts on super spreading and also on subcritical zoonoses and what goes on at the animal-human interface. Then most of the talk will focus on work I've been excited about recently, trying to sort of unpack the elements of transmissibility in collaboration with laboratory scientists. And some of this has to do with transmission experiments among animals, and then increasingly lately on virological experiments. So focusing on questions like viral stability in the environment, how that can be connected to transmission risk, and then a current major study on the effective temperature and humidity on viral stability. And then at the end, I'll quickly remark on this question of susceptibility in the novel host population or in the human population. I'll show case studies from monkeypox and influenza, and then remark on applications to COVID and possibly beyond. Okay, so transmissibility. It's the fundamental problem in infectious disease dynamics, and it's something I've been thinking about since I was a graduate student. I was midway through my dissertation when the SARS epidemic happened. Now we have to say SARS-1. And I was fascinated by the important role played by super spreaders. So I ended up digging up a lot of different data sets, beautiful contact tracing data sets for, I think, 10 different pathogens, and showed at the time that there was substantial individual variation from rare super spreaders at one end to non-transmitters at the other. And this appeared to be effectively a ubiquitous pattern in infectious disease. And one way of characterizing it is to think about what is the proportion of transmission contributed by the top 20% of spreaders. And this curve here shows that quantity as a function of the dispersion parameter. This analysis is all built on branching processes and negative binomial distributions. So the important thing to note is that as that dispersion parameter K gets smaller, there is greater individual variation in transmission. And you can see SARS is toward the extreme here. It's actually in excess of the famous 2080 rule. But all the pathogens that we could characterize are scattered along this curve, and none of them look like a homogeneous situation where everybody contributes the same amount to transmission. In that old paper, I went on to introduce a stochastic modeling framework. This is the branching process framework to analyze the consequences of this variation, and showed that greater individual variation means that any given introduction of the pathogen is more likely to die out, but that major outbreaks, when they do occur, tend to be explosive. And minor outbreaks, the ones that do die out, can be highly variable on the way to dying out. So it introduces a lot of noise at that point. So everything old is new again. So now we've got COVID happening, and it turns out that COVID exhibits a lot of super spreading behaviors as well. So a number of groups have estimated the dispersion parameter from COVID data, and it's found to fall in the region of SARS. You can see it's in around the 2080 point on that curve. And I think it's fair to say that each of those qualitative observations have been borne out for COVID dynamics as well. So we do see that many introductions into towns or other scale populations, many do die out, but some cause tremendously explosive epidemics, which are typically traced back to super spreading events. There are studies about this around the world now, but also that minor outbreaks are highly variable. So you can have significant flare-up in a region that is actually subcritical, and so it comes under control rapidly. But that gives rise to a lot of noise or variation in the range of outbreak size, even in subcritical regions. So that brings me to my next topic of thinking about subcritical spread. And it's important to note that all this super spreading work depends on having contact tracing data. which is beautiful, but is extremely rare. And in the world of pathogens at the animal-human interface, the data are typically much more degraded and challenging to work with. So one key question as we think about subcritical zoonoses is trying to disentangle, of all the cases you see, which of them come from zoonotic spillover versus human-to-human transmission. And so here's the cartoon of how little spillovers happen. Red cases are primary human cases, and they may cause little chains that die out. The challenge is, of course, that we don't see the transmission connections. We don't know, therefore, who is primary and who is secondary. And indeed, we often don't see some of the cases, so the data are degraded. We may not have any information on the reservoir itself, and so we end up working with a considerably less informative data set in terms of the total number of cases through time than the full mapping we would get from contact tracing. So how to approach this challenge? It turns out that this problem, this is a result from a paper that's now a bit old, 2009, a group of intrepid postdocs assembled all the models that had been done on modeling zoonotic pathogens at that time, and effectively found a big gap right at the animal-human interface, right where the zoonotic action is, meaning that at that time the field had not really grappled with the problem of teasing apart the animal and human contributions to emerging zoonotic spread. So in the decades since then, our group and a number of others have sort of dove into this problem. And we've come up with 3 new methods that use attainable data and address some of the common challenges about imperfect case detection or missing data of various kinds, and shown that this actually is a solvable problem. So this plot shows that we actually can estimate R, the reproductive number, just as well using fairly commonly available cluster size data, so the sizes of the clusters of cases that arise from spillover events, as we can using the gold standard contact tracing data. And then on the vertical dimension, we can do almost as well on the dispersion parameter, but there is some loss of resolution there. So this is a sort of best case scenario where you do have perfect case detection, but it shows what's possible and also opens the door to correcting for imperfect case detection. In another study, a recent student of mine, Monique Ambrose, introduced the spatial dimension using case onset data in data sets where we have place names but don't know how those locations are linked. So she estimated transmission parameters for a range of assumptions about the spatial scale of mixing. And the findings illustrate the fundamental trade-off between attributing cases to spillover or human-to-human transmission, depending how geographically widespread you allow that human-to-human transmission to be. The standard approach has been to assume that all human-to-human transmission is local, and so clusters are defined as occurring within, say, a village or town. But this actually yields a lower bound estimate of the reproductive number. And applying this method to monkeypox data from the Congo Basin, Monique found that the actual level of mixing seemed to be closer to the district level, which actually added a factor of 1/3 to the estimated reproductive number in this case. So those are some approaches to estimating transmissibility in this subcritical context. Another gap which our old review identified was in using models to inform how diseases should be controlled at this interface. And here, a current student, Riley Mumma, has done a lovely study, essentially trying to give model-based guidance for how to prioritize control measures versus either zoonotic spillover or human-to-human transmission. And this is really guided by this simple plot, which shows the expected proportion of cases coming from a human source as a function of the human-to-human transmissibility of a pathogen. So in the subcritical range, this expectation just goes linearly with the reproductive number, where as you move up toward 1, more and more of the cases are expected to be coming from humans. And in the endemic range, of course, all of the transmission is coming from humans. So what we can do with this is construct a model to think about the total incidence of of infection in a realm where there's a fixed level of spillover. And the black line here shows that that total incidence grows non-linearly as the reproductive number approaches 1 from beneath. That's as each chain on average gets longer. And then the colored lines show the effect of applying measures which reduce either spillover transmission or human-to-human transmission, or both if you're so lucky as to have a vaccine or something, of imposing a 50% reduction on each of these and seeing what effect it has on total incidence. And so what you find is that reducing spillover, you're just reducing the number of sparks, and that has a linear effect on the total incidence. That helps you a lot when R is low, but helps you less on a relative basis when human-to-human transmission is higher. In contrast, reducing that human-to-human transmission has a nonlinear effect, so it really begins to kick in and help you a lot as transmission is dominated by human-to-human spread. If you can reduce both, of course, that's always the best. But interestingly, when we analyzed a reactive policy where you focus on spillover and then switch to reducing human-to-human spread once an outbreak is discovered, you see that that's almost as good. And those 3 lines just show different delays in identifying and switching your control policy. That's 1, 2, or 3 generations of spread. And you can see they're almost as good as that purple line. So this sort of defines a space. You can see that the optimal measure clearly depends on how transmissible the pathogen is. And we can map that space out as a function of the amount of resource that's available and on the relative cost of the different measures. So here, moving from the bottom to the top, you're effectively saying you're moving from a low resource environment to an environment where you've got, unlimited resources, and you can effectively quench whichever type of transmission you're focusing on. As you pour more and more money in, it's always better in the end to focus on spillover. So the graph is fully orange at the top. But when you have limited resources, where you should focus your control measures depends critically on both the transmissibility, so how much of the transmission is driven by human-to-human spread, and on the relative cost of the 2 measures. So this defines a sort of rational basis for addressing those types of questions. Okay, so now I want to move on to thinking about laboratory studies and how we can learn more about transmissibility from all the excellent work that's being done in virology labs around the world. So I actually came into this thinking about pathogens that had not yet had any impact on humans. So when new viruses are discovered by either genetic surveillance or gain-of-function experiments or what have you, can we estimate their ability to infect humans or transmit among humans from indirect evidence? And the real origin was back in the day when those gain-of-function experiments were done on H5N1 influenza. There was controversy about whether we had created the very thing we feared. There were questions about whether these ferret transmission experiments actually gave us useful information about human-to-human transmission. And so what we did is we put together all the information we could find on ferret transmission experiments using a range of different influenza strains. And these were all strains that had been found in humans, but represented flu subtypes that were both seasonal, so supercritical, and strictly zoonotic, so subcritical. And this plot shows the result, the proportion of ferrets infected in various experiments, this mapped out by whether the strain involved was subcritical or supercritical. And you can see that there is a clear relationship there. And ultimately, we came up with this rule of thumb that if more than 2/3 of your ferrets get infected, based on these patterns, the strain is likely to be supercritical. And this is actually a stronger pattern than I expected to find. I came into this wondering if we would debunk the merit of ferret transmission as a metric for human-to-human transmission. But we ended up concluding that ferret experiments actually are a valid approach for screening influenza strains for pandemic potential, but of course that corroborating evidence is needed. And here the figure shows we actually got the data from those gain-of-function strains that spurred the controversy. And you can see that each of those is in the sort of red zone where our best estimate would be that that strain actually is supercritical in the human population. Note, however, the very wide confidence intervals there, which arise from the small sample sizes and the binomial uncertainties that arise when you're just saying 3 out of 4 ferrets got infected or what have you. So there are a number of big caveats and challenges with this type of approach. One being, of course, that transmission is a tremendously complex phenotype, and out-of-sample prediction is inherently risky, so corroboration is always needed. Another, though, is that for good reasons, these experiments use small sample sizes, and they typically just report it as a proportion infected. So there's a lot of, there's not much information there in that small sample size, and I would say much of it is sort of wasted in that simplistic analysis. Furthermore, we're getting just a crude prediction here of whether a strain is subcritical or supercritical. So we've gone on and done a further analysis to do a cross-scale model for these types of experiments. And here you see this is a typical data set from an experiment showing the viral titer by day for both source animals and the contact animals who ultimately do or don't get infected. And the objective here is to use the within-host viral titer data on the contact animals to get a higher resolution picture of transmission. And so you can, here's a little cartoon. If you sort of think about the viral titer that is measured in a contact animal, and you assume that that viral load grows exponentially in the early phase of infection, then you can estimate when the infection occurred rather than simply whether it occurred. So you're pulling more information from each given experiment. So if we then broaden that cartoon, here's a hypothetical experiment with 6 transmission pairs. Looking at it the conventional way, you would just say 4 out of 6 contact ferrets got infected, and that's the information you gain. But if you use this approach and estimate when exactly they got infected, then you end up with a richer data set with the time of infection across that set of ferrets, from which you can estimate a continuous quantity, the hazard rate of infection or force of infection, which gives more information about how transmissible exactly the virus was in that setting. So we did this using an MCMC approach to jointly estimate both the within-host growth rate and the force of infection by imputing the time of infection for each ferret pair. And as a test case, we analyzed all the data held by the CDC on ferret transmission experiments, grouping them by subtype. And the takeaway from this is that you get a better resolution in your insights about transmission than using the conventional proportion infected. And so in this bottom plot, you can see on the left, the different subtypes are separated in the vertical direction, showing the force of infection can tell not only that most ferrets got infected, but how quickly they did. Whereas simply thinking about the proportion infected, all the data are crowded together. And as a side benefit, we get a joint estimate of the within and between host fitness of these viruses, enabling us to begin to ask questions about how dynamics actually play out across scales. So this is quite preliminary, but I think promising in terms of the additional information we can gain from these hard-won data if we apply sort of model-based analytic techniques. A further frontier is to go deeper into the virology and actually to use models of the intracellular life cycle or the within-host, either straight-up kinetics or the spatiotemporal dynamics of viral spread within a host, to attempt to link together insights from all sorts of virological assays, in vitro experiments, in vivo growth, and even animal transmission experiments, to try to understand how all that knowledge coming from biomedical research in virology can be applied and synthesized to attempt to predict fitness outcomes that we care about at larger scales. So can within-host fitness be predicted from in vitro virological assay data, or can transmission be predicted from things like within-host fitness, tissue tropism, and other traits that can be measured in the lab? So I do think this is an exciting frontier, and had actually begun to dig into it with a wonderful group of people studying bat-borne henipaviruses. But as we were stepping up this effort, COVID happened, and everything stopped and changed. And so this particular effort got put on hold, and we pivoted toward COVID. But it was very fortunate because we had built these relationships with virologists who were working with already with dangerous bat-borne viruses. And so we were ready to hit the ground running in thinking about applying these types of ideas to SARS coronavirus 2. And so I want to tell you about some work we've done on virological measurements of SARS-CoV-2, in particular its stability, and how we link those to thinking about its transmissibility. So the initial question was, how long can this virus remain infectious in the environment? And this determines the risk from certainly from fomite spread if it's on a surface, and also potentially from airborne exposure. So our virologist collaborators at the NIH Rocky Mountain Labs, this is a group led by Vincent Munster, they put live virus on various surfaces and sprayed it into a mist and measured how long they could still detect live virus afterward. And then our group, which was folks in my lab at UCLA, and also Dylan Morris, who's a wonderful student who was finishing his dissertation at Princeton, but will be joining me soon as a postdoc, and I was able to sort of work with him in advance of that wonderfully. We worked together to model the data to extract the robust findings, given the data set, which was limited in scope, because this was all being done at BSL-4 conditions in the full sort of moon suit precautionary setting, and so they were not able to generate a lot of replicates, and so we had to squeeze all the information we could from the data that were available. So the mathematical model here is very simple, just an exponential decay model, but it's fitted using Bayesian regression, which accounts for a range of challenges we encountered with the data. So the raw data are shown here. This is a comparison of SARS coronavirus 2 in red and SARS-CoV-1 in blue. And you can see this is just the raw titer data testing the infectious titer of these viruses over time, both in aerosols in a rotating drum and across a range of different surfaces. These are the raw data showing what was measured over times up to 96 hours. This is how it was reported in the press. And this work was picked up very widely and with great interest, and many people asking how long they should leave their cardboard boxes on the porch and so on. But we were all frustrated that it was depicted in terms of, oh, it's 24 hours on cardboard, it's 3 days on plastic, because of course that depends how much you started with. Instead, the approach that we took was to model the decay rate of the virus, a viable virus, in each of these contexts. And then this in turn enables us to estimate the half-life in each of these environments. So here you see the posterior distributions on half-life of both SARS-2 and SARS-1 in aerosols and across this range of surface conditions. And so right away, this gave some actionable intelligence in terms of what are the approximate timescales we should be thinking about possible fomite risk or possible aerosol risk, absent all the other factors that we know apply to airborne transmission. So this paper came out back in March, I think. Immediately afterward, we pivoted to trying to broaden these findings on aerosol spread to think about the risk of airborne transmission. And at the time, as everybody knows, this went through various stages of being a relatively outsider or controversial view, and now it's increasingly accepted as we all deal with the patterns in front of us. But we dove into it because it was clear that this initial study focused on a very refined, a very singular part of the process, simply how long the virus could survive on a suspended aerosol. It did not deal with other factors that are crucial in the real world context, like the settling of aerosol particles and ventilation and things like that. So we wanted to try to put the pieces together to make a more practical assessment of the factors that linked this viral stability work to actual transmission risk. So this is more work led by Dylan and Ahmadine Gambal, who is a great postdoc in my group. It's remains unpublished because we dove into it and then we're distracted by the project I'll tell you about next. In any case, what we thought through was for an airborne infection event, you effectively need three processes to occur. You need creation of aerosolized virus, you need that virus to remain viable and present in the air, and then you need a susceptible human to become exposed and infected. And so we drew on the literature and built a mathematical model which integrates the known factors, so things like the virus decay rate, which we measured in the previous study, aerosol physics, particle size distributions, and dose response theory. And then we renormalized the whole problem in units of ID50, so the infectious dose that gives a 50% chance of infection, to essentially wall off the unknowns. So these are things like the aerosolization rate. So at what rate does a person or a medical procedure generate aerosolized virus? And then crucially, what is the infectious dose needed to create a new infection? Those things are not known, and so we bundled them up and buried them in this one quantity in the model. To do this, we consider different scenarios of contamination of an environment and subsequent exposure, which together give rise to an exposure dose and a consequent risk of infection. So just a couple of examples, you can imagine a contamination where an infected person coughs in a room for 10 minutes, then leaves. And then a susceptible comes in and spends, say, 1 minute in the room or 5 minutes in the room without any PPE. And those two exposures will give rise to different doses due to the duration. So hypothetically, it might be 1 ID50 or 5 ID50s for the amount of virus that was put into the environment. Another example might be an intubation of a patient, a known aerosol generating medical procedure of a patient at peak viral load. So that may put a lot of virus into the air. If the exposure is then a susceptible without PPE, that could be, say, 10 ID50s, so a near certain infection for that person. Or if they're wearing an N95 respirator for the same amount of time, that'll knock it down by probably 2 logs, actually. I should have changed that number. And so you'd have a significantly lower exposure dose given that PPE. So we're working in this kind of normalized risk scenario in units of ID50. And so here you can see the initial results, and just right off the bat, ventilation falls out as the dominant factor. It dominates over the virus inactivation and particle settling elements of the equation. And so in these plots, the 3 different colors show different risk scenarios, that is different numbers of ID50 in the exposure dose, how those decay from a point source contamination, how those decay through time. And then the different subplots reflect increasing ventilation rate. So 0 ventilation, if you're in, say, a closed elevator, to the levels of ventilation that are typical for a home or a patient hospital room, or an operating room or negative pressure room where they're really moving the air very quickly. And you can see that this is just the viable virus load as a function of time. You can see it responds very heavily to the ventilation rate, which obviously physically replaces the air and moves the virus out. So we see ventilation is key, and that the initial contamination load, unsurprisingly, has a sustained influence on the load of viable viruses in that environment. So we take that load and then we run it through a dose infectivity model, which translates it into the risk of actually becoming infected given that exposure. And again, you can see with low ventilation rates, risk can be sustained over quite extended periods. So it can be a number of hours that the virus, enough of it remains viable and suspended to infect people for hours after the initial contamination event. But as you move toward higher ventilation rates, up to hospital settings, or the outdoor setting would also be toward the right here, you see that the risk falls off much faster, down to tens of minutes to negligible risk. You can actually use the model to compute the waiting time needed to reduce the risk to some specified level. So for instance, if you think about what we call the medium risk event, which is an initial contamination of 10 ID50s, suppose you want to wait for risk to drop to 1 in 1,000 chance of getting infected. In an operating room with 12 air changes per hour, you would need to wait 30 minutes for risk to drop that low. In a patient room, you'd need to wait 2 hours. In an enclosed elevator or a car, it's up to 16 hours. So ventilation, again, is truly dominating this. Though it's important to note, again, that using PPE modulates what that initial risk level is for the exposure. And so that's a very effective way to shorten these waiting times. Okay, so as I said, that work got put on the shelf because we pivoted to another problem and another data set that we found very intriguing. And this is work on the environmental effects or the effects of environmental conditions on the stability of this virus. So it's known as a generality that the stability of viruses is affected by temperature and humidity. And this has long been thought to be one factor contributing to the seasonality of respiratory viruses like influenza and the other endemic coronaviruses. Though, of course, there are very important and possibly dominant other factors like contact patterns that change seasonally or other host factors. At the same time, it's clear that dependence on temperature and humidity will govern infection risk in particular environments, both indoor and outdoor environments. You know, the risk from a given exposure on a given day will be mediated by the survival of the virus in the environment that it finds itself in. However, the effects of temperature and humidity are not well understood, either in general or for SARS-CoV-2 in particular. And so this work, which again is with Dylan Anamandine and Kuei Yinda, who's a virologist at Rocky Mountain Laboratories, and their entire team, this work seeks to first empirically quantify this and then to model it to understand the general principles underlying it. So the experiments measured the stability of SARS-2 on plastic surfaces across a range of temperatures and humidities, so a full factorial mix of 3 temperatures and 3 relative humidities. And then we modeled it to try to extract the maximum insight again from these fairly sparse data and to seek generalizability. So just to show you the empirical patterns that come out, here you see for the 3 temperatures and across a range of humidities, you see the posteriors on the half-life that was measured. And the first thing I'll say is that it's not a small effect. So the half-lives vary by 20-fold across these measured conditions, which were not all that extreme. And even further, the right-hand points there reflect the estimates from the transient phase before the deposited droplets had fully evaporated, which we approximated as 100% humidity. And there you see even longer half-lives, although not very precisely estimated from these data, and an even greater relative effect across these environments. One takeaway point is that higher temperature always causes faster inactivation, which is sensible. But perhaps less obvious is that the effect of humidity is not monotonic, and indeed it exhibits a consistent U shape, which initially mystified us a bit. But then we began to look in the literature and find that this was a consistent pattern that has been reported over the years for different viruses, which got our curiosity up. So we have come up with a mechanistic model for the temperature and humidity effects here. And the premise is that the inactivation of a virus is driven by chemical reactions, which involve unknown reactants, but will follow the rules of chemistry. So the temperature dependence is governed by the Arrhenius equation. This is something you may recall from chemistry class years ago. And that's relatively straightforward, off-the-shelf stuff. The humidity dependence is more complicated. And here we're inspired by a group by Lindsay Marr, or work by her and her group a number of years ago, which divides the humidity response into 2 regimes, divided around what's called the efflorescence relative humidity. And this is the point below which the electrolytes in a solution will effloresce, which really means they'll come out of solution and form a crystal at equilibrium. Whereas above this efflorescence relative humidity, or ERH, you never you don't evaporate off all of the water, so the reactants remain in solution. And what changes is the concentration of those reactants, which will depend on the humidity, where in a more humid environment, there's more water and hence lower concentration. And so this gives rise to the qualitative expectation that reaction rates and hence the half-life will be relatively insensitive to humidity below the ERH, and then we'll have an increasing relationship above the ERH as shown in that diagram. So we took this and encoded it in a mathematical model by applying Arrhenius type kinetics both below and above the ERH, so in the crystal and solution phases of the reaction. And there are a few more parameters. The one important addition here is that above the ERH, there's this factor involving the S's, which is the relative concentration of the reactants in the solution as a function of the humidity. And the important thing to note here is that using this model, we can fit or predict the rate of the reaction, which is given by those little k's, using just 4 free parameters across the full range of any temperatures or humidities. So the blue circles show the parameters that need to be estimated. The green shows that the relative concentration as a function of humidity is something which you can measure directly in the lab just by looking at how much fluid has evaporated. And so applying this model to the data which were collected for SARS-2, and on the left you see data during the transient phase when evaporation is still ongoing. On the right you see after evaporative equilibrium is reached and the inactivation speeds up quite a lot. What you see is that we can accurately estimate the slopes for all 18 of these conditions, 3 temperatures, 3 humidities, transient, and equilibrium, all using just 4 free parameters. So the model does quite a good job of capturing all of the variation across these conditions. And then we can use it to actually create a sort of model-based representation of that non-monotonic U-shaped dependence of the half-life of the virus on the relative humidity. And here, those lines show the predictions of the mechanistic model across temperatures and humidities. And the points, those are taken from our data, but they're model-free estimates, so they're just regression fits. And it shows that the data broadly conform to the predictions of this mechanistic model. But that's an in-sample prediction, so it's not perhaps very impressive. We went on to do out-of-sample predictions. So this plot shows, again, the model is used to create the heat map, which is predicting the half-life of the virus across a range of temperatures and humidities. The big circles, the 9 of them, show our data, but the other shapes and smaller circles show data taken from the literature. So this is fully out-of-sample prediction of the half-lives, where we've just used the model fitted to our data and predicted what should the half-life be for, even for other viruses like SARS-CoV-1 or MERS. We're able to get quite accurate prediction of what their half-life should be, both above and below the ERH. And then we can go further. There are more data available if you use information from bulk media, where you can't estimate humidity, but you can do temperature. And there, if we normalize to a reference point within each of these studies, we're able to accurately predict the out-of-sample stability for 5 coronavirus species, including 2 endemic coronaviruses, across 5 logs of half-life. So this is going from minutes to multiple days, and spanning basically the full range of possible temperatures and humidities. So the takeaway from this is that we have found strong dependence of the stability of SARS-CoV-2 and other respiratory viruses on temperature and humidity. We measured half-lives from about an hour to about a day, and then we predicted half-lives over sort of reasonable conditions from half an hour to several weeks, which is a dramatic range. From this, we can think about transmission risk across environments and see that it will be greater at cool temperatures and at either low or high humidities, and intermediate humidities are worst for the virus. So this provides a mechanistic basis to think about some of the hotspots of transmission that have been observed. So meatpacking plants are cold and very humid, so these are perfect environments for the virus to persist and remain infectious. On the other hand, airplanes or some other controlled indoor environments tend to be cool and dry, which are also conditions which favor the virus. And this may be a contribution to future seasonality of this pathogen, though, as we know, the pandemic is largely blasting through seasonal variation at this point due to high susceptibility. In terms of the modes of transmission this work pertains to, it was measurement on surfaces, but we believe that it has implications beyond simply fomite transmission. And that's because there are similar qualitative patterns reported for aerosols and for smaller droplets. So the same U-shaped dependence on humidity has been observed. And so we feel that the same qualitative predictions and the range of sensitivity of half-lives to these conditions wouldn't carry over quantitatively, but the same patterns would be expected to carry through for airborne transmission. And crucially, a major source of airborne risk in some settings is resuspension of viruses from surfaces. And so the duration that viruses remain viable on surfaces is a crucial component of that risk. Finally, we feel that we put forward a mechanistic explanation for this non-monotonic effect of humidity. And the fact that we could do accurate out-of-sample prediction for other human coronaviruses suggests that the models picked up some general mechanisms which may govern the viability of enveloped respiratory viruses in general. Okay, so I'm on to the third part of the talk, but I think I am out of time. So I don't know if the moderator wants to, I can skip through to the very end and allow discussion, or I can cover, I just have a couple of slides on this topic, which I can cover quickly.
Do you want to do a couple of slides? And so, okay, I'll give you the option. You sacrifice questions or you do these slides. Which would you prefer?
I'll fly through in a couple of minutes very quickly, because for the most part, people can read it elsewhere. I just wanted to get these ideas forward and then we can get on to questions. So this final topic is susceptibility of the novel host population. For a long time now, I've been thinking about human monkeypox, which of course is a relative of smallpox, and the incidence and the age range of people affected by human monkeypox have both been rising steadily since 1980, which is the time when smallpox was eradicated and smallpox vaccination campaigns were ended. And so the graph here shows in the 1980s, the sort of age distribution of vaccination rates and how that has shifted by the 2000s, such that you have, entire cohorts of younger individuals who are not vaccinated. And then this pattern bears out directly in the epidemiology of monkeypox in humans, where both the age range of cases and the absolute incidence in a given region, which was surveyed in both periods, both of these have grown in proportion to the increasing fraction of the population which is susceptible. So effectively, by ending smallpox vaccination, we've initiated a demographic wave of susceptibility against other orthopox viruses which were predicted by that vaccine. So that's one example of what seems to be a virus that's novel to humans, or at least not widespread in humans, that is monkeypox virus, having non-trivial patterns in human population immunity which affect its epidemiology. Another, which we uncovered a few years ago, is influenza. So it turns out that immunological imprinting to your first lifetime infection with a seasonal flu ends up giving you lifelong protection against novel flu viruses, zoonotic flus, from the same hemagglutinin group. And I won't go into the details, this is all written up, but effectively past pandemic shifts in seasonal flu leave an imprint on the population scale, which gives rise again to demographic waves of susceptibility. And the plot there shows that you can predict the age distribution, which in fact is the birth year distribution of H7N9 or H5N1 cases from the history of influenza circulation in the human population. If this immunity is structured by age, strictly, then it will alter the transmission dynamics and the health impacts of a pandemic. So that figure shows two hypothetical H5 and H7 pandemics and how they would have different impacts across age because of this imprinting that has been measured. If the age-structured immunity is actually driven by birth year, so it's about when you're born and perhaps your initial flu exposure or your vaccination history, this actually gives rise to cohort-based waves of susceptibility in human demography, which give us opportunities to forecast emergence risk spanning decades using information that we have. And so that plot shows how the effective reproductive number of an H7 or an H5 might vary over decades as a consequence of continued exposure of cohorts to flu from one of these two hemagglutinin groups. That's all I'll say. You can read all about it in that paper. So the conclusions from this are that although these are viruses that are novel to the human population, the human population is partially immune. And this is arising from cross-immunity from the broader community of pathogens. And here I include the virus which is used as a vaccine against smallpox. This influences outbreak dynamics and health impacts and can give rise to strong age-based patterns and susceptibility. And in the event that they're actually driven by birth year, this gives rise to demographic waves which enable us to predict risk over time. So COVID-19 famously has differential impact across age, and it's interesting to think about whether these patterns from monkeypox and flu pertain to the COVID setting. So I think many of these points do apply for COVID, but it is not the same situation entirely. So it does appear that the human population has some pre-existing immunity. This is likely from the broader community of pathogens and exposure perhaps to other endemic coronaviruses. It has influenced dynamics and health impacts, and it's strongly age-driven. It is not at all clear, though, that it's driven by birth year, so when you were born. And in fact, it's impossible to discern whether it's driven by age or birth year, since all of our data comes from 1 year, and those two drivers would amount to the same statistical pattern. But all the available evidence suggests this is actually an age-based pattern, perhaps mediated to some degree by exposures of children to coronaviruses, or by entirely different factors like age-based expression of the viral receptor or apoptotic mechanisms or all sorts of other hypotheses. However, I do think it's important to note that for this present pandemic, it is another circumstance where the broader community of pathogens has left the human population with some footprint of immunity, which is shaping in a way that we don't yet understand how the pandemic is proceeding. I'll skip over this so we can get to our questions. Summarizing very quickly, I showed you some new techniques for estimating human-to-human transmissibility in difficult settings. I showed steps towards unpacking virological components of transmission and our attempts to use models to reveal general principles underlying these patterns. And then I quickly talked about how population immunity can be important even for pathogens that are novel to humans and can, for some cases, shape outbreak dynamics and emergence risk in predictable ways over a long period. So I've got some discussion starters, which I'll skip, but I do want to acknowledge the many people who've worked very hard. I tried to acknowledge them along the way, and of course our funders. So thanks for your attention, and sorry to go a little bit over time.
That was fantastic, Jamie. Thank you so much. I'm going to leap straight into questions because of time. So the first question was from Chris Dye, who asked, COVID super spreaders, do you think it's physiology, by behavior? Is it environment based on your last few slides? Do you have thoughts about that?
My thought is that I don't think it's simple. And I think, as always with super spreaders, it's multifactorial. So there is evidence of meaningful variation in viral load and viral shedding by individuals. And some of this seems to be, for SARS-CoV-2 in particular across humans, there are multiple log differences in viral load and presumably in shedding. Some of this is more general. So it's been known for some time that different people due to their own airway morphology or what have you, are better or worse at generating particles in the size range that can carry viruses efficiently. And then, of course, there are differences in behavior and mask wearing and what have you. So I think, maybe this is a boring answer, but I think that it's sort of perfect storm phenomenon where you have people. I think the best super spreaders are those with the physiological traits which enable them to create the right droplets for any virus, but also the immunological or physiological bad luck to have a very high viral load. And of course, this is all exacerbated by the pre-symptomatic shedding of SARS-CoV-2, which makes people more likely to be out circulating, attending weddings or going to bars or what have you, during the period that they're shedding a lot of virus because they don't feel it yet.
And you made it very clear that contact tracing data is really what we need. I wondered if you had thoughts about some of the shortcuts to getting contact tracing data, like apps, or do you think that's a hopeful direction?
I think it, whether it's, you know, the modernistic apps and Bluetooth tracing, or whether it is the boot leather, you know, hard work of contact tracing that's being rolled out at unprecedented scales, at least in some countries. I think there's tremendous scientific opportunity there to learn more about super spreading. You know, I've been thinking and talking about this for 15 years. And, you know, I think we've been hamstrung in our ability to really drill down into what drives super spreading because we haven't had high resolution data. We can sometimes get the data on the overall pattern, sort of post hoc, and we can build these network diagrams and estimate K parameters and so on. But in terms of understanding what drives it, which is what opens the door potentially to targeted control, having these high resolution data sets with linkage to the important biological and social covariates would really be a huge opportunity to advance the science. Now, of course, there are, as always, big privacy concerns, which, you know, have to be respected. But I have guarded hopes that there will be that the whole science of super spreading can take a sort of quantum leap forward in light of these huge data sets that are being developed and the new technologies.
Fantastic. So I'm going to ask one more question and then we can maybe move into the other session. And the question is from Brian. And he said, on the lovely humidity stuff, have you thought at all about summer viruses? So enteroviruses, have you thought about measles? Is a surprising question from Brian.
A little bit. Like I tried to be clear that we certainly do not believe that the environmental dependence of virus stability is the dominant driver of seasonality for any virus. You know, it probably does contribute, but we believe, and I should say I'm not personally expert on seasonality of endemic viruses, but on the whole, it seems likely that shifts in human contact behavior, you know, gathering indoors versus outdoors, possible changes in human physiology or immunity are likely to be, I would assess them to be the dominant factors driving seasonality of respiratory viruses. And measles, of course, is linked to school terms, as you have shown. So I think of this environmental stability work, first of all, in providing quite useful and actionable information at the proximate level of how transmission risk varies across environments, you know, on a given day. And in terms of its impact on seasonality, you know, I do think that if you marginalize all the other factors, there is an important difference between a virus that has a half-life of a half an hour versus a virus that has a half-life of a week. As I think about my kids and the day that they can ever go to a playground again and touch the undersides of metal bars, you know, I do think that it has an influence on some component of transmission risk, whether you expect the virus to be able to hang out there for a period of days versus effectively being gone in minutes. So I'm dodging your question about enteroviruses and measles, except to say that I think there are, you know, it's clearly a multifactorial puzzle, and I would expect that to be broadly different. You know, other forces may dominate over this viral stability in shaping those patterns. And something else I would add is that, again, I don't know this, but we've restricted our discussion of these data to respiratory viruses, which have evolved to cope with the respiratory epithelium broadly, that environment, as opposed to, you know, an enterovirus. Who knows what other things it may have evolved due to the different environments that it naturally passes through.
Of course, I hadn't thought about that. Great. Well, I feel like that should probably draw to a close the Q&A session of the talk.